Hi, and welcome to Protein Synthesis by Danielle Holt, Kenya James, and Dejana Ravel. Okay, so there's the DNA. The DNA, how you doing, is made up from the nitrogen bases, phosphate, and sugar. The nitrogen bases are AT and C to G. The helicase is now unwinding the DNA strand, and we will determine which one is either the leading or the lagging strand, whichever one goes from 3 to 5 or 5 to 3, as they separate. Once the helicase unwinds the parental double helix, the molecule of single-strand binding protein then stabilizes the unwound template strands on the leading strand. And then after that, the leading strand is then synthesized by DNA polymerase 3 from the 5' to the 3' direction. Okay, now let's look at lagging strand synthesis. Molecules of the single-strand binding protein stabilizes the unwinding template strands again. Primase joins RNA nucleotides into a primer. The DNA polymerase 3 adds DNA nucleotides to the primer, forming Okazaki fragment 1. Okazaki fragments are named after the Japanese scientists who discovered them and have about 100 to 200 nucleotides long in eukaryotes. After reaching the next RNA primer to the right, DNA polymerase 3 detaches. Fragment 2 is primed, then DNA polymerase 3 adds DNA nucleotides detaching when it reaches the fragment 1 primer. DNA polymerase 1 replaces the RNA with DNA, adding to the 3' end of the fragment 2. DNA ligase forms a bond between the newest DNA and the DNA of fragment 1. The lagging strand in this region is now complete.
Oh no, there was a mistake. Teams of enzymes detect this mistake, and a nuclease enzyme cuts the damaged DNA strand at the 2 points and removes the damaged section. Repair synthesis by a DNA polymerase then fills in the missing nucleotides, and then DNA ligase seals the free end of the new DNA to the old DNA, making the strand complete. What is the fastest way to determine the sex of a chromosome?
I don't know.
Pull down its genes.
Uh-oh.
What did one cell say to his sister cell when she stepped on his toe?
I don't know. Tell me what.
Ouch, mitosis! How do you eat DNA spaghetti?
How?
With a replication fork.
Ah.